Hello everybody, today we'll be talking about applications, advances and challenges of CRISPR-Cas9 in antimicrobial. In order to give you some context on it, have you guys watched the recent movie Rampage starring Dwayne "The Rock" Johnson? In this movie, a scientist has been working with a gene editing tool CRISPR and developed a gene therapy that might one day treat human diseases. However, things did not turn out well, and an accident turned a gorilla, a wolf, a crocodile into huge ferocious monsters. In reality, possibilities of creating such mutated monsters is there, but we are still far-fetched from it, as there are still many genes that we do not understand. CRISPR allows scientists to delete, modify, or completely replace the gene, and is discovered from a naturally occurring enzyme in bacteria like microbes or archaeas. They use CRISPR as a defense mechanism against invading parasites such as viruses. When viruses invade bacteria, they inject their own DNA into the cell. The bacterial CRISPR system will use its Cas complex to disable the virus and keep a spacer sequence to be used as immune defense. Upon secondary exposure to the viral DNA, it can recognize and copy the intruder's DNA sequence into RNA, which forms a complex with Cas enzyme, allowing it to disable the invader's DNA precisely. Details on how the mechanism works will be discussed later in this presentation. Scientists are able to extract this defensive mechanism from bacteria and utilize it as a gene editing tool and formulate many applications from it, such as in antimicrobials. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, so it tells us about the two components of CRISPR. First of all, the repeats of DNA, which palindromic means, is a sequence of letters that read the same from left to right. These are interspaced and identical. The spacer DNA fills up the interspaces which are unique to each other. It is found that this DNA matches perfectly with the viral DNA.
When the virus injects its DNA into the bacterial genome, the spacer in the CRISPR system will allow a Cas complex to be released which will transcribe the DNA to make a CRISPR RNA in order to fight the viral DNA and break it apart. When the viral DNA injected does not match any of the spacers on the CRISPR system, it will create a different class of protein, known as Class 1 Cas protein, which will take the DNA in and break it apart and then copies it into the CRISPR system, which will be used for subsequent attacks. Scientists hijack this system to delete genes or embed new genes. So for this CRISPR system, it only has one Cas protein, which is Cas9. The sections that look like wings can cut DNA. They are also creating two long strips of RNA, the CRISPR RNA and the tracer RNA. The linear part is the spacer segment that match with the corresponding viral DNA. The tracer RNA holds the CRISPR RNA in place and the whole thing forms a complex to break DNA. Scientists modified the whole CRISPR RNA by adding our own sequence of DNA instead of the spacer segment, as well as connecting both the CRISPR RNA and the tracer RNA, creating a chimera. So if we got a DNA we want to cut, we create a chimera that will have the same corresponding part to the RNA. This will allow the DNA to fit into the CRISPR-Cas9 complex, which will cut, creating an inactive or broken gene. We can also add new genes. This is done by inserting a host RNA as it is being broken apart. This is essential information as it can insert antimicrobial genes to combat against microbial infections.
Thanks for watching. If you liked this video, please give us a thumbs up. Thank you.